reticulum, it's rough, so it makes a protein. And the cells who make more protein tend to have much more, rough endoplasmatic reticulum. For example, plasma cells, yeah? When we take a look at a plasma cell, you can see that it has this, nucleus. Around the nucleus, there is this pale area, yes?
Yes, Doc.
This is basically rough endoplasmatic reticulum. And the rest is cytoplasm, correct?
Yes, Doc.
Why? Because what does a plasma cell produce?
Immunoglobulins, right, Doc?
Correct, yes. Nice. So the next thing they write...
Doc, any reason why you just showed the plasma cells, Doc?
Yeah, because this is a picture, they keep showing it to you. And they tell you, oh, this patient has multiple myeloma, and they put you an arrow at the pale area, and they tell you, what is this? And you have to say it's rough endoplasmatic reticulum.
Doc, is there any way we can look at other cells where the rRNA is present? You're talking about the rough ER, right, Doc?
Rough ER, yes, not RRNA.
And that's any other cells you can see it, Doc, please?
Yes, neurons. So in the neurons, there is something called the Nasal Body, okay? The Nasal Body.
Nasal Body, yeah.
Yeah, so I just need to find it. Okay. So this is the Neuron Body, yeah? Can you see this dark area? Darkish area? This is Nasal Body. There is a lot of it, correct?
Yeah, but I thought it was supposed to be a little lighter shade, Doc, than the cytoplasm. Like in the, plasma cell.
Yeah, well, in the plasma cell, it's lighter, but these stains are different from each other. So the stain we use for the brain is different from the one we use for the plasma cell, for example.
Okay, but for USMLE, I should just remember the plasma cell. Okay.
Yeah.
I'll do, Doc.
Okay, so we have this Nasal Body. You can see it here. What is the function of it in the neuron?
It's same as the rough area, Doc. Nasal Body, same as.
Yeah, so basically it helps us to make neurotransmitters, yeah?
Okay, yeah.
Okay. So it's synthesis of secretory or exported protein that go outside of the cell. And something called N-linked oligosaccharide addition to lysosome and other proteins. This function I will be explaining in the cell trafficking today.
Okay, Doc.
So Nasal Body, which is rough endoplasmatic reticulum in the neurons, synthesize peptide neurotransmitter for secretion to be able to secrete them outside. Basically, mainly it's neurotransmitter. Neurotransmitter should go to the synaptic cleft. So how can they move from the Nasal Body, which is in the body of the neuron, down to the axon and to the synaptic cleft? The answer is with the use of kinesin. Kinesin, okay?
Kinesin.
Yes.
Okay.
And how can they move back if they move back? With dynein, yeah?
Kinase and dynein arms, yeah.
Yep. Okay, so free ribosomes unattached to any membrane.
Are we talking about a neurotransmitter, Doc, or why did you talk about dynein and kinase, Doc, right now?
Kinesin.
What is moving through it?
Yeah, so kinesin help to move the neurotransmitter from the soma of the cell to the nerve terminal. Yeah? Well, dynein, I just said it for completeness sake. If you want to move something back to the soma, you need to use dynein. Like herpes, for example, herpes virus. Yeah? Herpes virus, when it infects us, how it moves back to the ganglia? The answer is dynein arm. Yeah?
But, okay, Doc, but where does it reside, Doc? Is it residing in the terminal for it to go back to the body?
Yeah, for example, like, let's say these are the lips, okay? And this is the trigeminal ganglia. Trigeminal ganglia has the cell body, yeah?
Okay.
Okay, so somebody get the herpes here. Herpes is going to move retrogradely using the dynein arm. When you are going to get a reinfection, it's going to move back to the lips using the kinesin, yeah?
Oh, okay.
Got it?
Where is it stored, Doc? In the neural body, Doc?
In the neuron body, but what do we call a neuron? A collection of neuron body outside of the CNS. We call it ganglia.
Ganglia. Okay.
Okay. So the trigeminal ganglia in this case. So
got it.
Free ribosomes unattached to any membrane are the site of synthesis of cytosolic, peroxisomal, and mitochondrial proteins. So they can make so many other things, yeah? So the next portion here, we have an end-linked glycosylation can occur in the endoplasmatic reticulum, which will help to basically, how do you say that? It's like you want to order an Uber to go somewhere, you know? So you tell him, okay, let me just give you the orientation, okay? So you put this end-linked glycosylation, which is like you glycosylate one end, and it's a way of telling him, man, you need to go to the, for example, the lysosome, yes? So it's just a direction information given to the particle. So this is how, for example, this protein know that he must get destroyed or he must exit the cell. Yeah?
Okay, Doc.
So mucus secreting goblet cells of the small intestine and also an antibody secreting plasma cells are rich with rough endoplasmatic reticulum because they are rich with the proteins. So protein...
Okay, Doc. Mucus producing goblet cells and antibody plasma cells. So they can give a multiple myeloma and show the plasma cells and ask for the RER.
Yeah, can you tell me please, do we have rough endoplasmatic reticulum? I'm sorry. Do we have goblet cells inside of our stomach?
I think so, right, Doc? Yesterday we were talking about it.
No, it wasn't the intestine. Do we have goblet cell normally inside of our esophagus?
Is it in the respiratory epithelium, the goblet cells?
It is in the respiratory epithelium, but what about the esophagus?
Esophagus, mucus, maybe yes, though?
No, it's not. Well, here's the thing. You must know that it does not exist in the esophagus and in the stomach. If it does exist, think cancer, metaplasia, okay?
Got it, Doc.
Good.
And one more thing, Doc, this end-linked glycosylation, Doc, like how we see in collagen synthesis, Doc. So in glycosylation state, they are adding the end-link, Doc, because I know that after getting out of the cell, they are being cleaved, right? They're in extracellular space. The C and N terminal. So in glycosylation state is where they attach it, the cell attaches end-link to the protein.
I'm lost, man. Which topic are you speaking about?
Like I just remember in collagen synthesis, Doc.
Collagen synthesis. I was searching for it here. I was like, okay, this one. Okay, what about the collagen synthesis? Tell me.
So in collagen synthesis, Doc, after in extracellularly we cleave the N terminal and the C terminal, right, Doc, of the protein.
Yeah, that's correct.
So, but they never say where it is attached. So this is where it attaches, Doc, in step 3. In glycosylation, they attach the N terminal to it for it to come out and get cleaved, any protein.
So you are asking when do we put the N terminal and the C terminal, correct?
Yeah, Doc, because over here it says, right, Doc, end-linked glycosylation occurs in endoplasmic reticulum.
So the N terminal and the C terminal are added since the beginning of the synthesis of the protein. If you remember when we had this initiator for translation, I told you that the N terminal and the C terminal were added. So they are added as the first portion of the process, okay? So they are all from the beginning.
Oh, yeah, Doc.
Yep.
They remove it and then add a cap and a tail, right, Doc?
No, man, why a cap and tail? We are speaking about a protein, not mRNA.
No, in translation I'm talking about, Doc.
Yeah, no need for a cap. The cap is in the mRNA, not in the... you are asking the initiator. Okay. So the initiator for translation recognizes the cap and start to decap it and take it in, basically, and allow the beginning of the process of translation. Correct.
Oh, okay, doc.
Okay.
Ending glycogen.
Okay. So here we have smooth endoplasmatic reticulum. So when we speak about the smooth one, it's the site of synthesis of steroid. Well, can you tell me what is the function of a steroid inside of our body? I mean, steroid ring.
It's definitely to build a cell membrane, doc.
Okay, what else?
Steroids to make cholesterol, doc.
Yeah, well, steroid ring can be used to make fat, I agree. Also for the cell membrane, also to make hormones. Yeah?
Okay, doc.
Glycocorticosteroid, mineralocorticosteroid, sex hormones. Yeah?
Yeah, doc.
Vitamin D as well. Okay?
Okay, doc.
I know vitamin D is atypical. That's why I told you about it. So,
Okay, doc.
side of steroid synthesis and detoxification of drugs and poisons. So it lacks the surface ribosomes that makes it smooth. And it has some enzymes in it, like a glucose 6-phosphatase. And if you remember, it was the last step of creating glucose from glycogen or from other substances. So it was the last step of a glucogenolysis and the gluconeogenesis. You don't have to know this now. We did not do metabolism yet. Okay?
So is the location of last step in blood glycogenolysis. So making of glucose last step. Okay, doc.
Yes. So liver hepatocytes and steroid hormone producing cells of the adrenal cortex and the gonads are rich with smooth endoplasmatic reticulum. Why do you think so?
Because that's why they make the hormones, right, doc? The proteins and everything.
Well, which hormone?
Okay, 1 second, doc. Liver hepatocytes and steroid hormones producing cells of rich in.
Well, remember, the adrenals produce which hormone? Cortex.
Adrenal, they give us the aldosterone, the sex hormones, and the cortico this things, right, doc?
Well,
Adrenal cortex.
Yeah, the mineralocorticosteroid, the glycocorticosteroid, and the sex hormone.
Glucocorticoids and sex hormones. Yeah.
Yes. Okay, so these are the steroids. Good. And these comes from the adrenal. What about the gonads? They make testosterone, yeah? Or estrogen or progesterone. These are sex hormones which also contain steroid in them. Okay. So far we finished with the rough and the smooth endoplasmatic reticulum. Do not forget that the smooth endoplasmatic reticulum is also responsible for the process of detoxification, especially for ethanol, okay?
Detoxification.
Again?
Did you say detoxification? Yeah.
Yeah. So can you tell me what is the difference between ethanol, methanol, and ethynyl glycol?
1 second, look. Detox. So ethanol is a regular vodka, dog. Ethanol.
Mm-hmm.
Right? And methanol is present in the cleaning stuff, right? Or cleaning liquids.
Yep.
And which one was the last one you asked, though? Propylene glycol?
Yes.
Propylene glycol is in the antifreeze, though?
Yep, done. Good. So cell trafficking, Golgi is distributions. Well, you know this, what is Golgi Apparatus? I like to think about it as Amazon. You know this Amazon, the company, I mean.
Mm-hmm.
Yeah, so they are just take the product and they distribute everything to which state, which city, which exact location it should go to. Yes?
Mm-hmm. Okay, doc.
So the Golgi Apparatus is the cell trafficking center and it's for distribution inside of the cell for proteins and lipid from the endoplasmatic reticulum to vesicles and the plasma membrane. Well, let's take a look. Here is the nucleus. The nucleus sends mRNA to the ribosome. It gets translated, and we have a protein. Yes?
Yeah, Doc.
The protein will move from the rough endoplasmatic reticulum into the Golgi apparatus. The Golgi apparatus is going to check, what should I do with him? Should I secrete him outside? Should I keep him inside? Or should I destroy him? Okay?
Okay.
If he gets something from the outside, you send him again to Amazon through Golgi apparatus.
Okay.
Yeah?
Okay, Doc.
So it's the distribution.
So, the whole thing is a Golgi apparatus, Doc? I thought that was a rough ER.
No. This is the rough ER.
Okay, Doc.
Good. So it's a post-translational event. Yes? It's post-translation, after translation. Correct?
Yes, Doc. Yeah.
So they write, post-translational event in O oligosaccharide include modifying the N oligosaccharide on asparagine and adding the oligosaccharide on serine and threonine. So you are like, what is that? What does it even mean? Well, look, don't worry yourself about it a lot, but let's imagine that you have box 1, box 2, and box 3, okay?
Mm-hmm.
You want to send box 1 to New York, box 2 to California, box 3 to Texas. So how can they know that? Well, it's all about the stamp, yes? So how can you stamp a box inside of the body? Well, you cannot write a label, send it outside, or you cannot write a label, keep it inside. Instead, you have to do adding or removing of substances, mostly adding. So how to add? What do you add actually? There is something called N-oligosaccharide, which can be bound into, for example, let's say box 2. And if it is added to box 2, well, actually, let's make it this way. These are exact same boxes. I mean by what they have inside of them. They are exactly the same. But I want to send box 1 to New York, the second one to California, the third one to Texas. How to do that? If you get the N-oligosaccharide, bind it to the asparagine, which is located in box 1, that will be sent into New York. That's it. Okay?
Okay.
Also, if I take the O-oligosaccharide and bind it into serine or threonine, which is again like amino acids, yeah? In box 2, it will be sent to California. If instead of it I put the N-oligosaccharide, it will be sent in New York, yeah?
Yeah.
If I have this box, the third one, and I put on it the mannose 6-phosphate, okay? It will be destroyed by the lysosomes, okay?
Okay.
So far so good. If you add something, it basically helps it to traffic. It knows
so
what
do I need to remember the amino acids, Doc, and what, like N is for aspirin and O is for serine?
Not really.
And threonine? Okay.
Not really, but there is one thing you need to know. Mannose 6-phosphate
it's for lysosomes.
It's for lysosome. This is a must to remember. The other one, not really, because there is no disease about them. And anytime there is no disease about something, it's low yield. So,
got it.
But mannose 6-phosphate phosphorylation, there is a disease. That makes it high yield. Okay. So the next thing they write, endosomes are sorting centers for material from outside of the cell. Okay. Or from the Golgi, sending it to the lysosome for destruction or back to the Golgi for further use. So there is this something called endosome. Can you see the endosome? The endosome takes something from the outside of the cell, and they are like, nice, well, we got this. What should we do with it? Well, do you like it? Yes. We'll send it to the Golgi, and he knows what to do with it. Do you like it? Not really. Give him to the lysosome and he will kill it. Destroy it.
Okay,
Yes?
Doc.
Okay, so the endosomes is the first recipient of the external stuff that comes from the outside. Yes?
Okay, Doc.
Okay, can you tell me really quick what is the function of Golgi apparatus? And how he does the sorting?
Golgi apparatus is an organelle in the cell, Doc. After the processing in the rough area, the protein, it enters the Golgi apparatus, and Golgi apparatus decides if it has to be sent outside the cell, or is it useful, or if not, it's bad, and it can send it to lysosome for destruction.
Okay.
And what else can it do? Yeah, it does the packaging by recognizing the amino acids, like adding the oligosaccharides on it to see where it's going to send it. And if it doesn't like something, it will attach mannose 6-phosphate and send it to lysosome for destruction. And endosomes are organelles which actually take if there's any protein from the outside which is trying to come inside, will take it. And if it likes it, it will send it to Golgi apparatus. If it doesn't like it, then send it to lysosomes for destruction.
Okay, I have a question. How does for example, you told the Golgi is responsible for processing the proteins, and you said it can send them to the outside, keep them inside, or go send them to hell, basically to the lysosome to be destroyed. How he can send them to the lysosome?
By attaching mannose 6-phosphate.
Okay, good. So they write here about the disease, which is the high yield disease for this topic. And this is actually like, if you knew nothing from this page, you must know this disease. Okay? So this disease, you will be asked on it. It's eye cell disease, also known as an inclusion cell disease or mucolipidosis type 2. So it's a lysosomal storage disorder, autosomal recessive disease, and the defect is going to be in an enzyme. I never was able to memorize its name. I recognize it by shape, but that they don't ask about the enzyme, they ask about something else. But you need to be able to recognize it, not memorize it. So the name of the enzyme, N-acetyl glucosamine 1-phosphotransferase. You can see why I don't memorize it. So it's really long name. They don't ask you about the name, they ask you about something else. They ask you about the next point. So what exactly happened in eye cell disease? There is failure of the Golgi cells to phosphorylate the mannose residue. So there will be less mannose 6-phosphate on the glycoprotein. Okay. If that is the case, can you deliver the substances to the lysosome to be destroyed?
No, Doc.
Okay, you cannot. So they write enzymes will be secreted extracellularly rather than delivered to the lysosome. Make sense?
Makes sense, Doc. Yeah.
Lysosomal defect in digestion of the enzymes will lead to buildup of cellular debris inside of the lysosome, creating inclusion body, which means basically that inside of the cell there will be plenty of substances not getting destroyed, regardless of the fact that they should be, and that will create an inclusion body. Correct?
I'm not understanding. 1 second, Doc. I understand that mannose 6-phosphate deficiency problem, and it's the lysosome is not getting anything. So those enzymes are secreted extracellularly, then delivered to lysosomes. Until there, I understand, Doc. The next, after the arrow, can you repeat the sentence and explain that?
Here, yeah?
Yeah, until there, it's all good, Doc.
Lysosomes deficit in digestive enzyme. Look, the lysosomes are here, and they should normally degrade the substances sent to them. Are you sending anything to them now after the deficiency?
No.
Okay, you are not. So look, if you are not sending to them, what is happening to the substance? It's either going extracellularly or it's going inside of the cell. Okay? So if it's going extracellularly, that's a problemo. But the problem is, there is one more problem. See this guy? I mean, Yeah, the lysosome here, they are supposed to get enzyme to be able to destroy the substances inside of them. Well, yeah, that's true that we don't have many debris basically coming inside of them, but still, few will be. But are we getting the enzyme, the lysosomal enzyme, that are responsible for destroying it? In another way, where is the lysosomal enzyme produced? The answers in the rough endoplasmatic reticulum, then goes to the Golgi apparatus, but now they cannot make it to the lysosome, correct?
Yes, Doc.
So if you cannot make it to the lysosome, can you destroy the debris inside of the lysosome?
No, Doc.
Okay. So what will happen to the debris inside of the lysosome? They will accumulate, yes?
But that's the thing, though. The debris is not going to the, because of no mannose 3,6-phosphate, it's not going to the lysosome, right, Doc?
The proteins are not making it. The proteins are not making it to the lysosome, not the debris. Debris is garbage. It's other stuff inside of the cell, like byproducts, garbage.
Okay. Okay.
So they write,
So,
so buildup of cellular debris inside of the lysosome, which lead to inclusion body. Again,
yeah.
lysosomes deficit in digestive enzyme, no digestive enzyme, will lead to buildup of cellular debris in the lysosome. Good?
Deficient in lysosomes. Still not getting it, Doc. I'm so sorry.
Okay, again.
Lysosome deficient in digestive enzymes, buildup of cellular debris in lysosomes.
If we have a cell, and here we have a nucleus, here we have rough endoplasmatic reticulum, and here we have Golgi apparatus.
Right.
Okay, and here we have
lysosome.
Okay,
Right.
so the enzymes inside of the lysosome, where do they come from?
Okay. That I don't know that.
Well,
I know the amino acids from the Golgi apparatus come.
Yeah, well,
But you're talking about enzymes. Okay.
Basically, they are encoded in our genes, translated, then come back to the lysosome. Okay?
Okay.
What about the byproduct of the cell? Well, they come from everywhere. Yes?
Okay.
Just the garbage. So this is the garbage. So let's say now we don't have phosphorylation of the mannose residue. Will you have the lysosomal enzyme going from the Golgi apparatus to the lysosome now?
No, Doc.
Okay, no protein going there. What about the garbage? The garbage will still be able to go there. Okay?
Yeah, Doc.
It's not a protein, it's garbage. So there is only garbage here. Yes?
Yeah, Doc.
So you will be able to see it as an inclusion. Inclusion is like something like this. Yeah? An accumulation inside of the lysosome.
Okay, Doc.
Make sense?
Okay, Doc.
Can you repeat it for me?
Just give me 1 minute, Doc. I just want to read it 1 second.
Okay.
So, enzymes. Okay, Doc.
Good. Can you tell
Got
it
it.
back to me?
So I'm thinking, Doc, amino acids are different and the enzymes are different. They are both going from Golgi apparatus to the lysosomes with mannose 6-phosphate. Right, Doc?
Correct.
And the enzymes are used for destruction of the protein, amino acids or any debris which is collected from all around the cell in the lysosomes.
Mm-hmm.
So since the enzyme is not making it, those bodies are retained in the lysosomes and they form the inclusion bodies.
Okay. So no lysosomal enzyme, but the debris are still there. Yes?
Yeah, debris is dead, that forms the inclusion bodies.
Okay, good. So they're right. So what are the symptoms? Well, the symptoms are actually quite weird. You feel like the pathogenesis is something, the symptoms is something else, but it's okay. They result in something called coarse facial feature. You know what that means?
No, doc.
You have to see it. You cannot, it's difficult for somebody to explain it. You have to see it.
Uh-huh.
It's like, exactly, you cannot describe it. Yes, this guy, or yeah, this guy. Okay?
Okay. I don't understand what's the deal.
Well, look, compare this with this. The nose is much more wider. Yes? The jaw
Okay.
is much more wider. Correct?
Okay.
And that happens within a year. That's coarse facial feature. This boy. Can you see?
Doc, when we spoke about potters, right, Doc? Is there also, do we see the name coarse over there? Like because of the less amniotic fluid?
Botter sequence. Well, let me remember. Not sure. I think so, actually. I think so. But basically, coarse facial feature means like the nose is wide, the jaw is wide. But I'm not sure about Potter sequence. You need to check that
I'll
one
just
out.
I'll just take a look at that 1 second.
Yeah.
No, nothing. Coarse. Okay, so this is the only place I will probably see. Okay, Doc. Coarse facial features.
Yes.
Okay.
Can you see the nose? It's wide. And I want you to notice the I don't know how do we call this area of the nose, but this area of the nose will be quite wide. Can you see it?
Okay, Doc.
Like normally in us, it's palpable, it's small, but in this patient, can you see it's flat? Can you see?
Yeah,
it?
Doc. Got it.
Yep.
Got it, Doc.
Good. So the next
So-
thing is coarse facial feature, gingival hyperplasia. Which medication lead to gingival hyperplasia?
I'm thinking calcium channel blockers, Doc.
Yep, and Infinity.
And phentoin. Phentoin.
What about anti-immune system drug?
Cyclophosphamide or something?
Cyclosporin.
No. Cyclosporin. Okay.
Yep. Corneal clouding, and that can lead to basically visual disturbances. Restricted joint movement, claw hand deformity, kyphoscoliosis. You know what kyphoscoliosis mean?
Yeah, Doc. The spine is sideways, right, Doc?
Well, when we say the patient has kyphosis, it's like this, yeah? But when we say he has a scoliosis, it's when he goes sideways, yeah? So kyphoscoliosis is like this and sideways, okay?
Okay. Can you show me, Doc? I'm sorry.
One moment.
Like in a picture. Kyphosis.
So this is kyphosis. Can you see it?
Okay, Doc.
This is scoliosis. Can you see it?
Sideways. Yeah.
Yeah, so this is kyphosis, this is scoliosis. What about kyphoscoliosis? It's when both of them are there. So let me find it. To get in the same time, when these two occurs at the same time. Yeah?
Okay, Doc. Any reason why this happens?
Well, one of them is this one, but to be fair with you, it's most of the time, it's because of disc herniation. I mean, in the normal population. You know?
Okay, Doc.
So this disease, we said something here. There was no phosphorylation of the mannose residue, and they were going outside of the cell, correct?
Yes, Doc. That's causing this. Okay.
So the lysosomal enzymes in the plasma, it will be increased, correct?
The lysosomal proteins. I thought the bad proteins, Doc,
The
which
what?
are not being destroyed. Okay. Yeah. I understand that.
So it's often fatal in childhood. How can they ask you about this? They can describe eye cell disease and they ask you why it happens, and it's failure to phosphorylate the mannose residue. Okay?
Got it, Doc.
That's the answer.
They won't give you the defect, Doc. The NA.
No, they don't ask about the enzyme. Never ask about the enzyme.
Doc, is there any short form for it? NA something, NAP or something?
No. It's like this. He's bulky. I don't memorize it actually. I don't like if somebody asks me about it, I will tell him I don't know. This is a problem in phosphorylation of the mannose residue. They can ask you at which organelle it occurs. This is a tricky question.
which organelle this occurs, Doc? Probably lysosome.
Ah, see, they tricked
Oh,
you.
no,
They tricked
it's in
you.
the Golgi because
In the
there's no
Golgi.
six. Yeah.
Absolutely. So this is a tricky question. So
Yeah.
the next thing we have is signal recognition pattern, which help us move to point A to point B again. So it's abundant cytosolic ribonucleoprotein that helps in the trafficking of polypeptide ribosomal complex from the cytosol to the rough endoplasmatic reticulum. What does that even mean? Well, basically, it helped to move the proteins from the cytosol to the rough endoplasmatic reticulum. Why would I do that? Well, you need to move some proteins to the rough endoplasmatic reticulum because eventually, actually peptide, because eventually you would need to disassemble them, get yourself some amino acid out of them, and rebuild other proteins from them. So the single recognition pattern are important for proteins to be how do you say that? Withdrawn from the cytoplasm. Also, the last thing you want is a dysfunctional protein accumulating in the cytoplasm. Correct?
Okay, then. Abundant cytoplasmic ribonucleoprotein that traffics. Doc, this is nothing to do with rRNA, nothing, right, Doc?
No. We are speaking about the proteins already.
Okay, Doc.
So this is
I'm not understanding this paragraph. Sorry, signal recognition particle.
Okay, so the signal recognition particle, it's a particle that is responsible of withdrawing bad proteins from the cytosol back to the rough endoplasmatic reticulum, leading to them being recycled. Okay?
Recycled, okay.
Yep. So if you don't recycle them, they will be accumulating inside of the cytoplasm, which can lead to problems. Correct?
Okay, Doc.
They don't ask about this. They ask always about ICEL.
Okay, Doc.
So the next topic is about vesicular trafficking proteins. These are kind of important. So if you look at the Golgi apparatus, it has two portions. It has something called the cis particle and the trans particle. Can you see it? So this is the cis and this is the trans.
Okay.
So vesicular trafficking proteins, they are called COB1. Well, it's COB1, COB2, but for me, I just make it easier for myself. I say it's COB1, COB2. So COB1, it exists in the Golgi, and the function of the COB1 is basically for retrograde movement. Look, can you see the COB1?
Yeah, Doc.
It helps to move the stuff from the cis portion of the Golgi apparatus back to the endoplasmatic reticulum, correct?
For recycling, Doc?
Yeah, exactly. Because sometimes you have some like amino acids or proteins that, well, they are not doing their job. So you would like to send them back to the endoplasmatic reticulum to do something about that. Yeah, basically makes different protein. Okay, so if we speak about the COB2, it's different. So they write, this is the COB2. Can you see it?
Yeah, Doc.
It's sending the stuff from where to where? From the rough endoplasmatic reticulum back to the cis Golgi. Yes?
Yes, Doc.
So COB1 sends back, COB2 sends forward. I like the mnemonic, it always helped me. 2 steps forward, 1 step backward. Okay?
Got it, Doc.
So, okay, so Cleritrin, one more guy. So the Cleritrin, it's related to the trans Golgi, and the trans
Doc, it's not Cleritrin, Doc. It's Cleritrin.
Cleritrin?
I thought Cleritrin is like an antibiotic, right?
Oh my God.
Clarithromycin.
Oh my God, because I just, I was speaking about this with somebody like 2 hours ago. So, okay, I get it. So Cleritrin.
Cleritrin, yeah.
Yeah, so it moves the stuff from the trans Golgi to the lysosomes. Okay? Okay, only not really. The Cleritrin also does much more than that. Notice, please. So it's moving the stuff from the Golgi apparatus to the outside. Also, it can move it to the lysosome. Can move it to the lysosome. Also, it can move it to the endosome basically, and then to the lysosome. So notice, this guy can lead to movement from the trans Golgi to the lysosome, or to the plasma or it can come from the plasma membrane. And you remember what I told you about the function of the endosome? They take the protein to the endosome. What does the endosome do?
It either sends it to the lysosome or back to Golgi Apparatus.
Okay, good. So yeah, or to the endosome, which is important for the receptor-mediated endocytosis. What is receptor-mediated endocytosis? For example, let's say we have here an LDL receptor, okay? If it recognizes an LDL, it's going to intake it inside, yeah? After taking it inside, it's going to the endosome with the help of the Cleritrin, yeah? So, and after that, they will decide, should we use it, keep it, or should we destroy it? Well, mostly it will be beta oxidation, so which is destruction, yes?
Okay, Doc.
Okay.
So, can you once again explain Cleritrin, Doc?
Again?
Can you once again talk about Cleritrin, Doc?
So yeah, Cleritrin helps for the cell trafficking again, and it can move between so many structures. For example, it can move stuff from the trans area of the rough endoplasmic reticulum to the lysosome for destruction. It also can help them to go to the endosomes, which will be then, well, either sent to the
I get it. Doc. It's between the plasma membrane endosome and between the trans portion of the Golgi apparatus to the endosome, to the lysosome. Okay.
Yeah, but that's not only. Also, Cleritrin can help with the process of endocytosis, which is intaking substance from the outside to the inside. So
Doc.
There is receptors that lead to formation of this vesicle.
Doc.
Yeah. Okay. All making sense?
Perfect, Doc.
Okay, good. So let's take a look about the next thing. We have the Berexus Arms.
Okay. Doc, can we take a quick break, Doc?
Yeah, sure thing, man.
Can you upload